morning, everyone, and I'm going to give a quick talk on whether we can restore Florida's inshore coral reefs using tolerant symbioses. The association between corals and the symbiotic photosynthetic algae from family Symbiodiniaceae can influence a coral holobiont's overall physiology, but more importantly, that coral holobiont's response to stress. Under temperature stress, a dynamic association, which is one where a coral can modulate the identity or the abundance of that symbiont, can contribute to a coral's survival. Much research has found that increased proportions of the genus Durasdinium within corals can increase a coral's survivorship under chronic bleaching stress. Moreover, the prevalence of Durasdinium species within Caribbean coral taxa has been increasing in recent years. This is likely a result of rising ocean temperatures driving environmental selection of this genus in the region, and suggests that its dominance in many coral species is a mechanism of coral bleaching tolerance. Within nearshore patch reefs along Florida's coral reef tract, the branching coral Acropora cervicornis was historically present, but is now locally extinct in a few habitats such as Newfound Harbor, which is south of Big Pine Key, and this is probably due to annual bleaching stress. All restoration practitioners that have previously used field nursery Acropora cervicornis outplanting at this shallow reef site, all outplants fail to survive after 1 year. However, most of the field nursery Acropora cervicornis contains the lesser tolerant Symbiodinium fiti, whereas at Mote Marine Laboratory's land-based nursery, sexual recruits have been found to contain Durasdinium, and this was identified through 2B rad sequencing. So last year we conducted a really small scale pilot study in order to basically track and investigate the symbiotic dynamics across natural heat stress to understand the symbioses and whether it can be influencing survival at these challenging reef sites. We outplanted 15 genotypes of batch-crossed sexual recruits. They are from 2018. We outplanted them from the land nursery to this shallow reef habitat in April, and we tracked the symbiont composition at 1 month post-outplanting during bleaching. We had recovery, and then a final time point when all temperatures had subsided. DNA samples were sequenced using the ITS2 molecular marker, and sequences were processed in SimPortal. To first orient you to the results of this study, they're very simple, straightforward, but it's a really beautiful story, or it's a very clean story, should I say. The proportion of the major ITS2 sequence is going to be on your y-axis. Each row is a unique coral genotype from the land-based nursery sexual recruits, and each column represents time, where that first column is going to be the ex situ, or that baseline symbiont composition. Then we have 1 month bleaching, recovery, and a final time point in the fall. If you do not see any bars for a particular time point or a genet, that means that there was no viable or living tissue remaining. That coral probably died. So first, surprisingly, even though 2B rad sequencing suggested that we had primarily Durasdinium in our sexual recruits at our land-based nursery, we actually had 5 of these 15 genotypes that contained Symbiodinium type A3. And they did not change their symbiotic association through the course of the study, where they actually bleached and then did not recover or survive. There were 5 other genotypes that also experienced bleaching during the study, but during recovery, they actually switched or shuffled to Durasdinium type D1, and they were able to survive the study. Lastly, we had the final 5 of the 15 genotypes. They either contained Durasdinium at lower proportions or higher proportions early on in the land-based nursery, or they switched or shuffled after 1 month of being outplanted, and they were completely unaffected by natural bleaching stress. So this very simple pilot study was kind of the characteristic example of the adaptive bleaching hypothesis, where symbiont shuffling or switching either before bleaching or during recovery can contribute to an outplanted coral's survival under natural bleaching stress. These findings present a novel and promising restoration intervention using bred coral recruits with tolerant symbioses, either those that occur naturally due to maybe nursery conditions or where you could experimentally inoculate these corals as they are devoid of any type of symbionts at their early stages of life during the rearing period. And that together, these tolerant symbioses have the potential to restore acroprid species at these shallow, challenging restoration sites. This study is ongoing. So this photo here is these corals are in their second year of outplanting. They've grown, they've fused, and they actually just experienced their second year of bleaching. And we do anecdotally see some bleaching, but we will see some survivors, and there are some that are also unaffected. Moreover, we're doing a full study this year looking at the potential for family contributions to symbiont fidelity, where those different patterns that we saw, those coral genets, were unrelated to one another, and so there could be a family contribution to your symbiont flexibility. In addition, we also have these coral recruits being reared in two different nursery environments, such as the land environment and the field environment, to see if there's any type of nursery or rearing environment influence on symbiont fidelity as well. If you have any questions, come find me later. Thank you.
We do have time for one
never
here
mind.
in the room, if anybody has any questions for Courtney. No? Actually, I do have one.
Yep.
How long do you think these associations last? Do you predict that year 2 and then maybe out 3 or 4, they'll maintain the Derstinium, or do you think they'll revert back to?
So we have samples from the 1 year time point, which was August of this year, and we have not done any sequencing. So our next question was, okay, when temperatures subside, are they going to revert back?
Right.
But are you giving Holland's talk?
No.
Okay. So we found that other acroporids maintain their symbioses many years following outplanting. So we're hopeful that that pattern is similar here in this case, but we will find out shortly with sequencing.
Okay, great. Thank
Cool.
you very much.